the year 1674, when the world was, well, it was literally about to double in size.
It's hard to overstate, really.
Yeah, and science at that point was largely the domain of elite academics, gentlemen philosophers who spoke Latin and lived within these university walls.
And that context, I think, makes our protagonist all the more remarkable. He was the ultimate outsider, a self-taught, often very secretive fabric seller from Delft in the Dutch Republic.
Antonie van Leeuwenhoek.
That's him. And he was about to, well, single-handedly launch the entire field of microbiology using this tiny homemade device.
A device so small, so simple, and yet so technically superior that the things he found, the secrets he uncovered, they were just immediately dismissed as fantasy.
Right. I mean, the sheer magnitude of his initial finding, just observing a microscopic darting creature, it was staggering for the time.
Is a discovery he communicated in a famous letter, right, to the Royal Society.
Yes, and we really have to appreciate the scale here. Imagine you're peering through this little piece of metal and glass at pond water, and you see, and I'm quoting here, a creature with green and very glittering little scales.
Wow.
And based on his really meticulous measurements, he estimated this little organism to be 1,000 times smaller than the smallest thing you could see with the naked eye, the mite.
1,000 times smaller. A whole new scale of existence, completely invisible but moving, alive.
Exactly. And that 1674 letter sort of introduced the concept, but the real explosion of knowledge came a bit later.
In the correspondence that microbiologists still refer to as just letter 18.
Simply letter 18. And that letter confirmed that this wasn't some, anomaly. Leeuwenhoek had started looking everywhere. In rainwater, in plaque he scraped from his own teeth.
From his own teeth, seriously.
From his own teeth, from pepper water. And he found what he called dierkens, or diertjes.
Which is just Dutch for small animals.
Right. And it was quickly translated into English as animalcules. But this wasn't just a list of new species. This was the monumental, world-changing documentation of single-celled organisms, of bacteria, of protozoa. He had revealed the microscopic universe.
So our deep dive today is going to focus on this incredible journey. We're exploring the unlikely life of this pioneer, the engineering genius, and of course the secrecy behind his powerful single-lens microscope.
A tool that remained unmatched for 150 years.
A century and a half. And we'll look at the intense skepticism he faced from the academic establishment and ultimately how his 17th century curiosity really set the table for the central unresolved questions of 21st century biology.
We'll be touching on massive concepts like endosymbiosis and the modern human microbiome.
It's really tracing the lineage of knowledge from a piece of curved glass held up to the light in Delft all the way to the complex chimeric nature of the modern tree of life. Okay, let's unpack this. This story is about a craving for knowledge that led a businessman to become the father of microbiology. Let's begin by grounding ourselves in the man and his environment. Antonie van Leeuwenhoek was born in Delft, in the Netherlands, in 1632. You really have to picture the scene. This is the Dutch Golden Age.
Right. It's a time of just profound innovation in both art and science.
And he was right in the middle of it.
Indeed. He was right in the center of this Dutch cultural and scientific ferment. And it's worth noting that he was a contemporary of the famous painter Johannes Vermeer, who also lived in Delft.
They were neighbors, weren't they?
They were neighbors, and Leeuwenhoek was involved enough in civic life that he was actually appointed the executor of Vermeer's will when the artist died so suddenly in 1675.
But unlike those high society academics of the time, Leeuwenhoek didn't come from a lineage of formal learning. His background was much more modest.
Oh, definitely. He was not part of the established academic elite. His father was a simple basket maker. His mother, though, did come from a somewhat better-off family of brewers.
So a bit of a mixed background.
A bit. But his early education was brief. His father died when Antonie was only 5, and he was sent off to school in Warmond. By the time he was 16, he was apprenticed as a bookkeeper and cashier at a linen draper's shop in Amsterdam.
And that apprenticeship in Amsterdam, that was really the turning point, wasn't it? That experience basically formed the foundation for his entire life's work.
It was absolutely crucial. After 6 years there, he returned to Delft in 1654, opened his own draper's shop, selling various textiles like linen, yarn, and ribbon.
Okay, so this is where the lenses come in.
This is where we assume they come in. It was common practice for drapers to use convex lenses, simple magnifying glasses, to examine the thread count and the quality of their fabrics.
Quality control.
Exactly. And this is where we think that initial practical spark for his obsession with lens making came from. He needed a tool for his business, and he became fascinated with the tool itself.
But his career wasn't purely commercial, though. He leveraged his meticulous nature and gained a pretty significant standing in Delft.
He did. His status grew, and he secured several important, well-paying civic roles. Starting in 1660, he took on the role of chamberlain for the city sheriffs.
And he held that post for nearly 40 years.
Almost 4 decades. And these duties were administrative, managing the premises, keeping things orderly, but they were also highly confidential. He was privy to sensitive city discussions, and his job required him to maintain absolute silence on what he heard.
That suggests a highly trusted and discreet individual.
Absolutely. And on top of that, he also served as an official land surveyor, and maybe most lucratively, he was the official wine gauger of Delft.
Wine gauger, what does that entail?
He was responsible for measuring and taxing all the city's wine imports. So these jobs, they gave him an income, stability, and maybe most importantly, the protected, quiet time he needed to pursue his real passion. The lens. And that passion wasn't motivated by, external rewards. He was driven by an almost internal necessity.
There's that great quote of his.
There is. He once wrote, "My work, which I've done for a long time, was not pursued in order to gain the praise I now enjoy, but chiefly from a craving after knowledge, which I notice resides in me more than most other men."
A craving after knowledge. I love that. And that innate, intense curiosity is the force that manifests as his secret weapon, the single-lens microscope.
And the word secret is key.
Because he kept the method for making his best lenses entirely to himself.
The secrecy was legendary. Leeuwenhoek dedicated decades to this. It's estimated he made over 500 optical lenses and at least 25 complete single-lens microscopes.
And we only know that because so few survive.
Right. Only 9 of those instruments survive today, serving as these sort of physical relics of his unparalleled skill.
When we talk about these microscopes, we really shouldn't picture the big, brass, ornate instruments we might associate with 17th century science.
No, not at all. Leeuwenhoek's devices were revolutionary because of their simplicity and their scale. They were minimalist to an extreme. People often describe them as tiny, thumb-sized pieces of engineering.
How small are we talking?
The largest surviving examples are only about 5 centimeters long. The instrument was basically just 2 flat, thin metal plates, copper, or, for his finer instruments, silver riveted together.
And sandwiched between them was the critical component.
The single handmade biconvex lens.
That sounds incredibly fiddly to use.
Right.
I mean, how did he manage to focus and navigate his samples with such a minimal device?
It was certainly challenging. The whole operation required holding the entire instrument right up to your eye. The design included this mechanism with a small metal pin where the sample, a drop of water, or a sliver of tissue was mounted.
And that pin had to be incredibly close to the tiny lens for focus.
Incredibly close, so you needed very fine motor control just to view the specimen.
And how did he adjust it?
Focus and navigation were managed by a series of 3 screws, which was really ingenious. These screws allowed the pin and the specimen to be moved in 3 independent axes. One screw adjusted the distance for focus, and the other 2 let him scan and navigate the area of the specimen.
It must have demanded immense patience.
Immense patience and manual dexterity. But the payoff for this difficult manual operation was just astounding performance.
Unparalleled, right?
Completely. While the surviving microscopes confirm a magnification up to 275 times, it's strongly suspected, based on the dimensions of the bacteria he described, that he had lenses that could magnify up to 500 times.
But as we've discussed, magnification is only half the story. The key was the resolution.
That was the real secret. Leeuwenhoek's microscopes achieved the resolution of less than 1 micrometer, 1 meter.
And to put that in perspective for you, the typical bacterial cell is around 1 to 5 nanometers long.
So his resolution allowed him to clearly define the structure of individual bacteria, something that was entirely impossible for anyone else at the time. This ability, seeing structures at the submicron level, was the secret to his success, and it remained unmatched for over 150 years.
What's fascinating here is Leeuwenhoek wasn't just observing. He was conducting these ingenious experiments with a curiosity that preceded any map or, established bearings for this unseen world.
Yeah. He was drawing his own map as he went. He wasn't relying on university texts.
Right. He was driven to create this technological leap that solved a fundamental optical problem resolution, which the entire academic community would fail to solve for another century and a half.
He was a true self-made pioneer. He didn't have the benefit of established scientific methodology, so he relied purely on empirical evidence and meticulous recording. That Innate, intense curiosity was coupled with the discipline of a meticulous lens maker and businessman.
Which allowed him to explore and manipulate this microscopic universe with these startlingly original experimental insights.
He had the power to see, and he was the first person to even think of looking that closely.
So with this secretive, powerful, tiny instrument, Leeuwenhoek starts documenting observations that justified all 17th century assumptions about life. What was on his list of firsts?
The list really, it marks the foundation of modern biology. He essentially established two new fields. He was the first to give detailed descriptions of protists, those are the larger single-celled organisms, and bacteria he found in various water sources.
The famous animalcules.
Right? His dear Akins. And his observations were truly universal. They spanned across multiple kingdoms of life.
It wasn't just pond scum.
No, not at all. He documented the structure of muscle fibers. He observed blood and was the first person to describe red blood cells, noting their shape and size in incredible detail.
And he even saw them moving.
He went on to document blood flow in capillaries, which finally proved the missing link in William Harvey's theory of circulation. And in a self-experiment of immense historical importance, he documented human spermatozoa.
Which immediately sparked one of the era's strangest debates about reproduction.
Oh, absolutely. And he even turned his microscope toward disease and pathology, didn't he?
Yes. He documented the crystalline structures found in gouty tophi. Those are the chalky deposits that form in the joints of gout sufferers. This again just showed his range. He applied his lens not just to novel life forms, but to human pathology.
The famous pepper experiment, I think, always stands out as this great example of science progressing through error.
Can you elaborate on that?
Sure. It's a classic anecdote that illustrates his hands-on empirical approach. Leeuwenhoek initially had a hypothesis that the spiciness of pepper must be caused by microscopic, sharp spikes.
That makes a certain kind of sense.
It does.
Mm-hmm.
So he soaked pepper in water, intending to look for these spikes. Of course, he didn't find any spikes, but what he accidentally did was create a nutrient broth.
Yeah.
And instead of spikes, he found the water was just teeming with tiny moving organisms, protozoa. His hypothesis failed, but the accidental observation led to a monumental discovery about the ubiquity of microscopic life.
This sounds like monumental, world-changing information. But because he was an outsider, writing in his native Dutch, this information transfer created a huge challenge, the communication paradox.
That's right. Leeuwenhoek was entirely outside the formal academic network. He communicated exclusively through letters, often rambling, chaotic, almost like a diary in his own colloquial Dutch. He never published a formal scientific paper in Latin.
Which was the lingua franca of European science at the time.
Exactly. So the Royal Society of London became his primary conduit to the world, but this required an enormous leap of faith and labor on their part.
So when he sent his first detailed letter describing single-celled organisms in 1676, the reaction wasn't immediate acceptance.
Not at all. It was met with severe skepticism, bordering on outright disbelief. You have to imagine receiving a letter from a Dutch draper claiming he could see creatures 1,000 times smaller than a grain of sand, moving and darting around in water.
Their existence was entirely unknown.
Completely. And while he had established a reputation for reliability through earlier, less fantastical observations, like capillary blood flow, this new claim just stretched credibility past its breaking point. They suspected he was either unreliable or, frankly, delusional.
So how did the Royal Society bridge the language barrier and this credibility gap?
They relied heavily on their secretary, a man named Henry Oldenburg. Oldenburg was so instrumental that he actually taught himself Dutch specifically to translate Leeuwenhoek's voluminous correspondence.
Wow, he learned a new language for this.
He did, just to convey the findings accurately to the wider scientific community. And Oldenburg also gave us the term we still use today. He translated dinoctins into the English animalcules.
And this leads us to Robert Hooke, the great English microscopist, author of Micrographia. Hooke had to be the final judge here.
Hooke's role was essential for validation. The Royal Society had a standard. Extraordinary claims require extraordinary proof. So they tasked Hooke, who was maybe the most accomplished microscopist in England, with reproducing Leeuwenhoek's observations.
And he did it.
He did. The Society only fully accepted the astonishing findings, and thus accepted Leeuwenhoek, once Hooke successfully replicated the experiments. At that moment, the self-taught Draper from Delft became an international scientific celebrity.
And Hooke, despite being a sort of rival in the field, was deeply respectful of Leeuwenhoek's skill.
Highly respectful. Hooke admired his dedication so much that he, too, taught himself Dutch to read the original letters. And in describing the reproduction of the observations, Hooke validated the relative sizes Leeuwenhoek reported.
There's a great quote from him about that.
Yes, he referred to the larger protozoa as gigantic monsters in comparison of a lesser sort, which almost filled the water, the latter, of course, being the bacteria. Hooke was confirming not just the existence of the creatures, but the astounding resolving power of Leeuwenhoek's instruments.
Here's where it gets really interesting. I mean, imagine being the first person to tell the world about life so small it was invisible, only to have your audience, the most learned men in Europe, question whether you were hallucinating.
It must have taken profound self-confidence.
And it had to be reinforced by that internal craving after knowledge to just keep sending those letters.
It speaks volumes about his conviction and the meticulous nature of his work. His results were repeatable, but only if you had the right instrument. And that's where the drama lies, in the fact that the established academic experts, using their preferred complex tools, they simply could not see what the Dutch Draper could see.
That whole scientific drama,
Mm.
the lone secretive genius versus the institutional establishment, it's central to the Leeuwenhoek legend. We've established that Leeuwenhoek's simple, single-lens microscopes were just technically superior to the more complex compound microscopes used by academic scientists like Hooke. But why? What was the physical, optical limitation that the academics failed to overcome?
The issue lay in the physics of light, specifically with errors that we know as refractive aberrations. When light passes through lenses, it bends. Compound microscopes rely on stacking multiple lenses to get higher magnification.
But the flaw is that when you stack lenses, at least in the 17th century, the distortions they compound. They get worse and worse.
Exactly. Can you break down what that distortion looks like?
Sure. The two main types were spherical and chromatic aberration. Spherical aberration means that light passing through the center of the lens focuses at a different point than light passing through the edge.
So the image is blurry and indistinct, especially towards the edges.
Right. And chromatic aberration is even worse. Since glass acts like a prism, different colors of light, different wavelengths, they bend at slightly different angles. So the image breaks down into these little rainbow halos around the edges of objects.
So the more lenses you added for magnification, the blurrier and more colorful the image became. The simple single-lens instrument just bypassed this problem entirely.
Exactly. Leeuwenhoek used only one incredibly small, perfectly ground biconvex lens. By using just one lens, he drastically reduced the compounding errors of aberration. He noted that his simple instrument provided objects that appeared much more clear and distinct.
He was solving a problem that physicists wouldn't fully correct until the achromatic lenses of the 19th century.
Which brings us back to the core mystery, his intense secrecy. He actively discouraged teaching his lens-making techniques, which guaranteed his unique advantage. Why was he so fiercely protective?
Leeuwenhoek maintained throughout his life that there were aspects of the construction which I only keep for myself. His motives were likely complex. I mean, they certainly included protecting his scientific priority, his income, but also maybe a sense of proprietary pride.
He did leave some to the Royal Society, though.
He did. When he died in 1723 at the age of 90, he showed immense generosity by gifting 13 of his microscopes to the Royal Society. But critically, the technical knowledge of how he achieved those perfect high magnification lenses, it died with him.
And that secrecy created this vacuum of knowledge that persisted for centuries. We only got a definitive clue to his method relatively recently.
Yes, just in 2021, a team of researchers in the Netherlands used some really cutting-edge technology to examine a surviving Leeuwenhoek microscope without destroying it. They used non-destructive neutron tomography.
Non-destructive neutron tomography. That sounds extremely high-tech. Why neutron specifically?
Well, neutrons are ideal for this because they can penetrate the dense metal plates, the copper or silver casing, very easily, but they scatter strongly off light elements like the glass of the tiny lens and any organic remnants of the sample.
So it allowed them to create a precise 3D map of the lens structure and its placement, which X-rays couldn't do because of the metal casing.
Exactly. And what this internal mapping revealed about his secret technique was fascinating. The analysis strongly suggests that his superior lenses were not conventionally ground and polished, which was the standard method.
So how did he do it?
It seems they were likely created by fusing thin glass thread, resulting in what's termed a blown glass lens, or a Stong-Masolov-type spherical lens. This method was easier for producing the tiny, near-perfect spheres needed for such high power.
And this reveals a fascinating, almost tragic irony involving Robert Hooke, the very man who validated Leeuwenhoek.
The irony is profound, and it suggests Leeuwenhoek's secrecy might have been motivated by protecting his reputation.
How so?
The very technique identified by modern tomography, creating high-quality spherical lenses by fusing thin glass threads, was actually first devised, described, and published by Robert Hooke in the preface to Micrographia in 1665.
No way. So Leeuwenhoek may have effectively borrowed, refined, and perfected a method Hooke had publicly shared and then abandoned, and then claimed the results as his own private secret.
That's the supported hypothesis. And adding to the confusion and the irony, Hooke himself strongly disliked using these simple, high-power instruments. He found them very troublesome to be used because they were so small and required holding a lens right up against your eye.
So Hooke's preference for the easier, larger compound microscope was passed down to the next generation of academics. And this preference for convenience over optical performance had devastating consequences for the field.
It was a critical point that led to the 150-year hiatus in microbiology. The entire scientific community adopted the inferior compound microscope as the standard tool, and the superb resolution achieved by Leeuwenhoek was just forgotten.
What happened to microscopy during this period of decline?
Well, the field lost all credibility. Because the compound microscopes produce these blurry, distorted images from those uncorrected aberrations, observers started reporting figures that were imaginary, or just artifacts of poor optics.
Like the homunculi.
The most infamous example, yes. The belief in homunculi tiny, fully formed humans crouching inside semen, which supported these preformationist theories of reproduction. The unreliability of the instrument led directly to the unreliability of the conclusions.
And if the observations were deemed unreliable, the subject matter itself was just dismissed.
Exactly. The invisible world of animalcules became a footnote. Even the great taxonomist Carl Linnaeus in the 1750s, he barely bothered to classify microbes. He just lumped them into a catch-all genus called chaos, meaning formless, within the phylum Vermes or worms.
So the idea that you could see genuine cells as small as bacteria was dismissed as a 17th century fantasy.
Right, and this scientific dark age only ended after more than a century and a half.
When was Leeuwenhoek finally vindicated?
The true vindication happened much later, in 1981, when a researcher named Brian J. Ford rediscovered some of Leeuwenhoek's original specimens in the Royal Society Library. Ford successfully photographed these original samples using a surviving Leeuwenhoek microscope, proving beyond a shadow of a doubt that the instrument could achieve a resolution of less than 1 micrometer. The records were true. The world has simply forgotten how to see.
This raises such an important question. I mean, what kind of scientific progress is lost or delayed when a key technical advance is kept secret, even if the intention is to protect one's work?
The move to the inferior compound microscope meant microbiology stalled for decades. A whole discipline was delayed.
It's this immense tension between scientific fame and communal advancement.
Leeuwenhoek secured his reputation forever, but his secrecy arguably delayed the greatest biological discovery, the role of microbes in life and disease, by generations. We see the inverse of that today, where rapid sharing is the norm, but the lesson remains. Technology is only useful if its methods can be shared and replicated.
Okay, so let's fast forward. The hiatus ends. The world finally catches up with Leeuwenhoek's resolution. And suddenly, his 17th century observations become precursors to the largest, most profound biological theories of the 20th and 21st centuries.
Right. One of the most fascinating details is his observation of globules within the larger animalcules.
This is from that 1677 paper.
Astonishingly prophetic. He was observing a large ciliate, a type of protist. We think it was Colpidium colpoda. And he noted that its body was filled with 10, 12, or 14 globules, which lay separate from each other.
And then he compared the size of these internal structures to the very smallest free-living creatures he'd seen, what we now know are bacteria.
Exactly. And those internal globules fit the dimensional profile of mitochondria. We're talking about organelles that are the powerhouses of the cell with a diameter between 0.5 to 1 micrometer.
Which is right at the limit of Leeuwenhoek's resolution.
Right at the limit. So if he was seeing mitochondria or chloroplasts, he was observing the physical evidence of endosymbiotic theory 250 years before it was accepted.
And this theory is foundational to our understanding of complex life. It proposes that eukaryote cells, like ours, with a nucleus and specialized compartments, originated when an ancestral archaeal host cell engulfed or acquired bacterial endosymbionts.
That concept that one type of life swallowed another type of life and they became one was initially ridiculed, wasn't it?
Famously so. Early 20th century pioneers like Konstantin Muraszkowski and Ivan Wallen, they championed this idea of symbiogenesis, arguing that mitochondria and chloroplasts were originally free-living bacteria.
And the idea was considered too radical, too fantastic for polite biological society.
It was only in the 1960s, largely through the work of Lynn Margulis, who provided overwhelming evidence from DNA sequencing, that the theory finally gained acceptance.
Leeuwenhoek, having faced his own lifetime of skepticism, would have understood that disbelief perfectly.
Beyond the origin of the cell itself, the evolution of all those animalcules needed a framework. And that framework came, ironically, from Leeuwenhoek's own hometown, Delft, from the work of Albert Kleiver.
Yes, Kleiver, who held the microbiology professorship in Delft in the mid-20th century, provided the philosophical underpinning for microbial life. He championed the fundamental unity of biochemistry.
So before him, the metabolic world seemed chaotic. Fermentation, respiration, photosynthesis, they all seemed like totally distinct processes.
Right. Kleiver realized that all these processes, across all forms of life, are driven by shared mechanisms, primarily involving electron transfer and phosphorylation to generate energy.
And this is where we get that famous, catchy axiom.
Precisely. Kleiver's original observation was that from elephant to butyric acid bacterium, it is all the same. This was simplified and popularized by French scientists into the axiom. What is true for bacteria is also true for elephants.
An insight that began to explain the evolution of Leeuwenhoek's bewildering variety of little animals. It showed that despite all their structural differences, the basic metabolic engine is universally conserved.
Exactly. Kleiver's unity of life paved the way for scientists to formalize the grand division in cellular architecture. Stanier and Van Niel in 1961 sought to define the distinction between the two types of cells Leeuwenhoek first observed.
And this is where they formally introduced the concepts of prokaryotes and eukaryotes.
Right. Prokaryotes, bacteria, and archaea, which lack a nucleus, in eukaryotes. Protists, fungi, animals, and plants, which have a nucleus. And their key insight was that the difference wasn't about function.
Because both prokaryotes and eukaryotes can respire or photosynthesize.
Right. It was about the organization of their cellular machinery.
In simple terms, how did they define that difference?
They argued that the prokaryotic cell is the indivisible functional unit. Everything happens in one main compartment. In contrast, eukaryotes rely on specialized membrane-bound compartments organelles like mitochondria to perform specific processes.
And this division set the stage for attempts to map the grand evolutionary tree of life using molecular data.
A quest that really started with Francis Crick advocating for protein taxonomy and was formalized by Carl Woese using ribosomal RNA.
Why was ribosomal RNA the initial tool of choice?
Woese used ribosomal RNA, rRNA, because it's a critical component of the cell's protein-making machinery, and it evolves very, very slowly. Critically, the gene that codes for it is usually inherited vertically passed directly from parent to offspring, making it a stable molecular clock.
And his work gave us the famous three-domains tree.
Yes, it showed that prokaryotes weren't a single group, but split into two deep domains, bacteria and archaea. And eukaryotes were placed as a sister group to the archaea.
But that single gene tree is now considered misleading, especially when we look at the whole picture provided by modern genomic sequencing.
Profoundly misleading. The issue is that the RNA gene represents barely 1% of a microorganism's total genome. Modern sequencing shows that eukaryotes are not simple branches off the archaeal trunk, they are genomic chimeras.
Meaning they're the result of that singular, ancient, endosymbiotic fusion of an archaeal host and a bacterial symbiont, the ancestor of the mitochondrion.
Which happened roughly 1.5 billion years ago. So instead of the tree dividing neatly, it fused. And that changes the very definition of an organism.
And it explains why complexity became possible.
It does. This endosymbiotic fusion, where the archaeal host gained the energy-producing capacity of the bacterium, resulted in an increase in available energy per gene of 3 to 5 orders of magnitude compared to their prokaryotic ancestors.
An enormous burst of energy.
Which overcame the growth constraints of bacteria and permitted the evolution of complex, energy-intensive traits like the nucleus, sex, and phagocytosis traits first observed in Leeuwenhoek's protists.
But the complexity doesn't stop with endosymbiosis. The microbial world also actively exchanges genetic material horizontally, which makes drawing a simple tree almost impossible.
That's the second equally disruptive factor, lateral gene transfer, or LGT. Unlike vertical inheritance, where genes pass down the generations, LGT means genes are constantly being swapped horizontally between unrelated cells, even across species and domains. Think of genes as an exchangeable currency in the microbial world.
So if 99% of a microbe's genome is constantly being swapped and transferred, then basing the entire evolutionary map on the 1% that's vertically inherited, like RNA, is fundamentally flawed.
Exactly. The constant lateral transfer of genes for things like antibiotic resistance or new metabolic pathways just muddies the deep phylogenetic signal you need to draw a unified tree. It's why some evolutionary biologists, like Ford Doolittle, argue that the universal tree of life is simply a human foible.
An attempt to force a messy, net-like reality into a neat branching diagram.
Right. And if we connect this back, Leeuwenhoek, without a map or bearings, was happily exploring a chimeric world where the rules of inheritance are constantly changing. The lesson is perhaps that biology, particularly microbiology, is still a land without a compass. We shouldn't trust the map if it doesn't reflect the complex reality of gene exchange and cellular fusion.
We need to embrace the fluidity.
Leeuwenhoek observed a teeming, dynamic world. The current molecular revolution just confirms that the most profound evolutionary events were not about slow branching, but about fusion and widespread genetic exchange. The microbial world is defined by constant collaboration and negotiation.
Leeuwenhoek's 17th century discovery of animalcules laid the ultimate foundation for the modern understanding of microbial life as essential collaborators. And today, that understanding has completely revolutionized how we view our own bodies through the lens of the human microbiome.
The current relevance is unavoidable. We are now acutely aware that microbes are the indispensable foundation of Earth's biosphere. They generate our oxygen, recycle essential nutrients. It makes perfect sense that life on that scale would be essential to our own existence.
So let's define the modern concepts clearly. The human microbiome and the microbiota.
The human microbiota is the collection of all the microbial residents, the bacteria, archaea, protozoans, fungi, and viruses that inhabit us. The microbiome is the collective genomes of those organisms.
And we now know that the typical adult human is host to approximately as many microbial cells as human cells.
Right. Which fundamentally challenges the notion of a single, defined human identity. It reframes us as more of an ecological habitat than a discrete organism.
Leading researchers to describe this collection as a virtual organ, or even the neglected organ.
And this microbial community performs tasks that we genetically simply cannot. It's been dubbed the second liver because of its crucial role in regulating host metabolism, breaking down compounds we can't, producing essential vitamins.
It's also called the second brain.
Because of the dense concentration of neurons in the gut, the enteric nervous system that communicate constantly with the central nervous system via microbial metabolites, and the collective genetic power of this community, it dwarfs our own.
Which is why it's also known as the second genome.
Absolutely. If you just consider the gastrointestinal tract alone, its microbiome includes at least 3.3 million non-redundant genes.
3.3 million. And to emphasize that for you, the human genome has about 20,000 genes.
So this microbial community comprises an astonishing 150 times more non-redundant genes than the entire human genome. This incredibly complex, integrated system means humans and their resident microbiota must be defined as a superorganism.
And this superorganism exists in a dynamic equilibrium. So what happens when that equilibrium is lost? This is a key concept in modern medicine.
The loss of equilibrium is called dysbiosis, and it is often triggered by external factors like dietary changes, stress, or most commonly antibiotic use. And dysbiosis is increasingly linked to a huge range of diseases, from obesity and autoimmune disorders to neurological conditions.
What are the three characteristic ways dysbiosis manifests itself in the body?
We generally categorize it into three types of community changes. First, you can have the proliferation of harmful microbes. A simple, visible example is dental decay, where acid-producing species like certain streptococcus thrive on sugar, ferment it, and break down tooth enamel.
Okay, and the second type involves the beneficial residents being pushed out.
Exactly, the depletion of beneficial microbes. A crucial example is the female genital tract, which is dominated by acid-producing lactobacillus species. They maintain a low pH that is inhospitable to opportunistic pathogens. When these beneficial lactobacillus species decline, the infection risk increases dramatically.
And the third type relates to the overall health and resilience of the community as a whole.
That's the loss of overall microbial diversity. A healthy gut microbiome is highly diverse. It offers redundancy and resilience. When that diversity decreases, for instance, a loss of the most abundant groups like bacteroids and firmicutes, that loss is a key characteristic of diseases like inflammatory bowel disease.
Which is concerning because those firmicutes are known for their anti-inflammatory effects.
Right, through the production of short-chain fatty acids.
So Leeuwenhoek used his tiny handheld lens to glimpse a few moving dots. Today, we map the entire superorganism using tools that process terabytes of data. The 21st century equivalent of Leeuwenhoek's high-resolution microscope is metagenomics.
And metagenomics is a revolutionary tool because it circumvents the fundamental limitation that plagued microbiology for centuries, the fact that 99% of all microbes cannot be cultured in the laboratory. By retrieving genomic information directly from entire microbial communities in a sample, say a stool sample, we reveal the true biodiversity.
We mentioned two main approaches, targeted sequencing and shotgun sequencing. Let's make that distinction accessible.
Okay, let's use an analogy. Targeted sequencing, often focusing on the 16's ribosomal RNA region, is like scanning the ISBN barcode on every book in a massive library. The barcode is unique to the class of book, the genus, or family of the microbe. It's fast, it's cheap.
But it tells you the identity of the organism group, not what the organism actually does or the specific strain it is.
Exactly. It gives you the author's name, but not the book's content. And shotgun sequencing.
Shotgun sequencing is the heavy artillery. It's like gathering up every single page from every book in the library and sequencing the text of all of them simultaneously.
This process non-discriminately sequences all the genetic material in the sample. And thanks to modern sequencing speed, this approach provides high-resolution taxonomic composition down to the species and strain level, and, crucially, it gives us functional annotation. We can map those 3.3 million non-redundant genes to specific biological functions, what the superorganism is actually doing for us.
So what does this all mean? We went from a single lens seeing one tiny animal to sequencing millions of genes in a single sample. But the goal, it remains the same. Mapping the territory Leeuwenhoek first glimpsed.
And the quality of that map is entirely dependent on meticulous technique, just as Leeuwenhoek's clarity depended on his perfectly ground lens. Modern metagenomics demands rigorous standards for sample collection, ensuring consistency, minimizing contamination. The technology has scaled unimaginably, but the need for Leeuwenhoek's meticulous observation and careful technique is mirrored in the modern genomic lab.
We began with a secretive, self-taught fabric seller in Delft in 1674, moved through centuries of scientific skepticism and delay caused by the lost secret of his lens, and we've arrived at the 21st century realization that we are complex superorganisms, shaped by a microbial second genome that governs our health, our metabolism, and our evolution. It is a breathtaking journey rooted entirely in the sustained curiosity of Antonie van Leeuwenhoek.
And while our computational power is immense, Leeuwenhoek's initial discovery set in motion these profound questions at the heart of biology that remain stubbornly unanswered today.
Like what?
Well, we've acknowledged that the origin of eukaryotes involved a massive singular fusion endosymbiosis. But we still know of no surviving evolutionary intermediates between the simple prokaryotes and the complex nucleus-possessing eukaryotes. The transitional forms are completely missing.
That is the central black box in the story of complex life. The singular event that birthed the architecture of the cell that defines everything from protists to elephants.
Exactly. And we still lack a consensus on the factors that drove the evolution of the basal eukaryotic traits that Leeuwenhoek first observed in his larger animalcules. Things like the nucleus, meiosis or sexual reproduction, and phagocytosis, the ability to engulf other cells.
So why did the nucleus evolve in eukaryotes but not in their archaeal ancestors? What prevents most bacteria from engulfing their neighbors? These are still wide-open, fundamental questions.
The technological leap from a 5-centimeter copper plate with a tiny lens to a next-generation sequencing machine is huge, yet the core mystery persists.
Leeuwenhoek was happiest without a compass in this new world of invisible life. We might think our metagenomic tools provide a complete final map.
But the continued prevalence of lateral gene transfer, where genes are swapped across the evolutionary landscape, and the fundamental unresolved mystery of complex life's origin show that we, too, are still explorers in the microbial world. We are mapping a territory that fundamentally resists being drawn as a simple tree.
The biggest question Leeuwenhoek might have asked remains, why is life the way it is?
And perhaps more pressingly, What are these constant genetic negotiations doing to the future of life on Earth?